if blood pressure changes, and remember, blood pressure is going to go hand in hand with blood volume, okay? Why? Because blood pressure is a hydrostatic pressure. It's the pressure that the blood exerts against the walls of blood vessels. So if blood volume decreases, blood pressure decreases. If blood volume increases, blood pressure increases. They go hand in hand, right? So if blood pressure or blood volume drops, all right, let's say you get a person coming to the ER bleeding out. They got a stab wound or were in a car accident and they're bleeding. So their blood volume is decreasing, which means that the blood pressure is decreasing as well. This decrease in blood volume and therefore blood pressure is going to be sensed by these specialized cells, the juxtaglomerular apparatus. You can call it just JGA, or you can call it glomerular cells. That's the other name they have. So these glomerular cells are specialized cells on the afferent arteriole. So you guys remember we have the afferent and efferent arterioles joined to the glomerulus, the glomerular capillaries. So this here is afferent. So blood goes here to the glomerulus, and this one over here is efferent arterioles. And these specialized cells here, the juxtaglomerular cells, they're also called, you can just call them granular cells. Granular cells. So granular cells or juxtaglomerular apparatus, either one, it's fine. But they themselves sense a decrease in blood pressure. Because what's going here? Blood, right? And those cells are going to sense that less stretch in that blood vessel or more stretch in that blood vessel. So if we have low blood volume and low blood pressure, these cells are going to sense that decrease in pressure, okay? The next thing that is going to happen is we have these other cells that you see right here, the yellow ones. And those cells are called the macula densa cells. Let me write down that better. Macula densa cells. And those cells are part of the distal tubule. So remember I told you that the nephron is kind of folded onto itself? This is how it looks. The Bowman's capsule is right here, and the distal tubule is next to it. So these macula densa cells are part of the distal tubule. And they're specialized. They're going to sense low filtration rate. Because remember what happens. If blood pressure is low, the capillary pressure in the glomerulus is also going to be low. So the capillary pressure is low. If capillary pressure is low, that is the force for filtration, and glomerular filtration rate is going to be low. If we're filtering, making less filtrate, so that filter is going to go into the proximal tubule, then the loop of Henle, which is not shown here, then the distal tubule, and these macula densa cells are going to sense that low flow because we have less filtrate moving through. So two things are happening here. The granular cells, or juxtaglomerular apparatus, however you want to call it, which are part of the afferent arteriole, are going to sense the decrease in blood pressure. Okay? The macula densa cells, which are those in the distal tubule, located right here in the distal tubule, are going to sense the low filtrate flow. Why? Because if blood pressure is low, capillary pressure is low, less filtration rate, less filtrate flow through the distal tubule, macula densa cells sense that, and they're going to signal these juxtaglomerular apparatus or granular cells to release renin. Okay, so these cells sense the signal from the macula densa and the low blood pressure, and they are going to produce and secrete renin. So those granular cells on the afferent arteriole are the ones that make renin. What is renin? Renin is an enzyme. It's released in the circulation in the blood, but it's not a hormone, it's an enzyme. What renin does is that it converts a protein called angiotensinogen, produced by the liver, into angiotensin 1. Okay? That angiotensin 1 is the inactive form of a hormone. Which hormone? Angiotensin 2. So when angiotensin 1 is circulating in the blood, it's going to encounter another enzyme called angiotensin converting enzyme, or ACE, and that converts it into angiotensin 2. And angiotensin 2 is a hormone that does almost everything in your body. Angiotensin 2 is going to cause vasoconstriction of arterioles. It binds to angiotensin 2 receptors on the smooth muscle of arterioles. It's going to cause vasoconstriction, which increases resistance, which increases blood pressure. Because remember, that diagram that I used to show you guys when we looked at blood pressure regulation. If you have here your left ventricle, right? Here you have the aorta. And here you have arteries and arterioles. I'm going to skip the arteries just to make it easier. If we cause vasoconstriction, so this lumen of this blood vessel becomes smaller, right? So the radius decreases because we vasoconstrict, we make the lumen smaller, then there's going to be less blood flow because we're increasing resistance, right? And if we increase resistance, we are going to decrease blood flow. So less blood will be able to come out here, right? So that's going to increase blood volume in the aorta, which increases blood pressure. Does that make sense? That's how we, by vasoconstricting arterioles, we're going to reverse the change in blood pressure. We're going to bring this blood pressure back up. The other thing that is going to happen is that angiotensin 2 is going to go to the adrenal and communicate with cells in the adrenal gland, specifically the adrenal cortex, which is the outside of the adrenal gland, and cause it to release aldosterone. So aldosterone is going to cause the reabsorption of sodium. Remember, reabsorption is bringing something from the filtrate back into the circulation, recycling it. So we're going to reabsorb sodium. If we reabsorb sodium, that's like the equivalent of eating a bag of chips. What happens when you eat a bag of chips? You're going to do two things. You're going to feel thirsty, so the hypothalamus will initiate that behavior of drinking fluids, and if you drink fluids, you increase blood volume, therefore you increase blood pressure. And the other thing that happens here is that water will be reabsorbed if we have antidiuretic hormone present. Okay, so water will be reabsorbed if we have antidiuretic hormone or vasopressin present. But it turns out that angiotensin 2 also goes and talks to the hypothalamus and posterior pituitary gland and tells it release ADH. And so that will initiate that insertion of aquaporin 2s in the distal tubule and collecting duct. And if we insert aquaporin 2s, we're creating a pathway for water. And we have the osmotic gradient of the renal medulla, so we can reabsorb water from the collecting duct. And we have that osmotic gradient between the filtrate in the distal tubule. Remember, that's 100 millismolar, and the renal cortex is 300 millismolar. So we have that driving force. We're inserting the pathway, we can reabsorb water. If we reabsorb water, we're increasing blood volume, therefore we're increasing blood pressure. Does that make sense? The other thing that angiotensin 2 does is not shown in this diagram, but angiotensin 2 goes and talks to the cardiovascular control center in the brainstem and increases that high sympathetic, low parasympathetic, affecting blood pressure regulation. Does that make sense, guys? So this is how the kidneys regulate blood pressure, through the angiotensin-renin-aldosterone system. Any questions? Okay. In my review session, I'll bring these together with the blood pressure regulation by the autonomic nervous system, and we'll review all these pathway. All right. I just wanted to put this here. So remember, antidiuretic hormone present, then we reabsorb water. If we don't have antidiuretic hormone, then we don't reabsorb water. Okay. Now we're going to move on to the digestive system. And these are the learning objectives of the digestive system. This you can use, I'll produce a study guide by Wednesday, but you can use the learning objectives to study for now. All right, so we're going to talk about diet first. An adequate diet must satisfy 3 needs, okay? We need to get chemical energy for cellular processes, for the cell to be able to do its work. We need to use these building blocks, right, that we get from things that we ingest in the diet to form macromolecules, okay, to make things like we need them for making membranes, for making enzymes, for making a number of different structures in our bodies. And then we need the essential nutrients that are the essential nutrients are required materials that need to be obtained from the food because our body cannot produce it. And they are things like essential amino acids, essential fatty acids, vitamins, and minerals. Okay, so these 3 things we are going to use. There are things that we get from the diet that we're going to need to have for our bodies to be able to create structures, repair structures, and do cellular work. What are the types of nutrients? So we have macronutrients. Macronutrients are going to be carbohydrates, fats, and proteins. So carbohydrates, when we break them down, as I'll show you in just a minute, are going to be converted into simple sugars, like glucose. It would be an example of that. Fats, they're going to be broken down into glycerol and fatty acids. And proteins are going to be broken down into amino acids. Okay, and these are the things that then we absorb. We move them back into our circulatory, or we move them to a circulatory system, and we can use them for the 3 purposes that I showed you in the previous slide. Micronutrients are vitamins and minerals, and these are also important. Vitamins are super important for maintaining certain things in your body, like, for example, vitamin E for collagen, stuff like that, vitamin D for bone. And then we have minerals like iron for carrying oxygen. It's the one that binds oxygen in the hemoglobin. Calcium, again, for bone, etc. All right, what are the states of nutrition? So we have these 4 states of nutrition. Ingestion is taking the food into the digestive system. So that's ingestion is going to happen here in the mouth. I'll talk about the anatomy of the digestive system in a minute. Digestion is the chemical and mechanical breakdown of food. So chemical by enzymes and other chemicals like or substances, like, for example, hydrochloric acid in the stomach. These, for example, bile. So we'll talk about all these things later on. But it's chemical, and then mechanical is, like actively breaking down by either chewing, as we will see in just a minute, or with muscle contraction that also helps mechanical breakdown of food. And why do we want to break down food? We want to break down food because these big macromolecules are not going to be able to be absorbed back into the circulation, right? We need things to be tiny so that they can diffuse. Remember, diffusion rate depends on how small the molecules are. So we need to break them down. Like proteins won't be able to be absorbed. We need to break them down into amino acids that we can then absorb, bring back into the circulation so that our body can use it. The digestive system is actually considered outside the body, okay? Absorption is when we have that those small food particles, so the digested particles, and they move across the intestine wall into capillaries, and then taken into the circulation, taken into the blood. And then elimination is it's excretion, right? It's the removal of waste from the body. Things that we don't need to absorb, we don't need them in our body, they're going to be excreted to the outside. All right, let's look at the anatomy of the GI tract. So first we have the mouth with the teeth and tongue and saliva. That's important. I'll talk about it in the next slide. We have the pharynx. The pharynx is a compartment that is shared between the respiratory and the digestive system. After the pharynx, we have the esophagus that connects that oral cavity with the stomach. The stomach is right here. Stomach is where we're going to have that hydrochloric acid. I'll talk about the stomach in detail in just a minute. After the stomach, we have the small intestine, which is the location where, or the compartment where most of digestion and absorption actually happens in the small intestine. After the small intestine, we have the large intestine, which mostly participates in water reabsorption. There's some digestion there, but usually by the microbiome. And then we have the rectum and anus for excretion of things that we don't want, that we want to get rid of. And then we have the accessory organs, which are the pancreas, you see it right here, and the liver. And here you have in green the gallbladder. And these are super important. I mean, you guys probably don't think about the pancreas very, very often, but without the pancreas, a lot of the digestion won't happen. Without the liver, a lot of detoxification of your blood won't happen. Lipid digestion won't happen. So these are very important organs. So they call them the accessory organs, but they play a key role in digestion of those macromolecules. All right, let's start with the mouth. So in the mouth, you're going to have mechanical digestion. So you have the teeth breaking and the tongue actually actively breaking down the food into smaller particles. And that's important because if you break down into smaller particles, you increase surface area. The surface area to volume ratio increases, and that helps certain enzymes to chemically digest more effectively those food particles. So mechanical and chemical digestion happen in the mouth. The chemical digestion is salivary amylase. You need to know that. You have in the saliva, you have this enzyme called amylase, and that enzyme breaks down carbohydrates. Okay, so you start digesting carbohydrates right in the mouth, in the oral cavity. So mechanically break it down so that the salivary amylase can now act more effectively, chemically break it down into smaller pieces. And then that's going to be swallowed. So here you have the epiglottis that when it's up gives way to the respiratory system. So when you're going to swallow, this epiglottis closes. So a little flap that closes and closes off the respiratory system so that food or drinks don't go in the respiratory system. It goes into the esophagus. The esophagus, which is that long tube that connects the pharynx with the stomach, has two sphincters. This is the upper esophageal sphincter. So once that opens, a sphincter is a ring of muscle that is normally closed, and it opens when something needs to go through. So this is going to open so that food can enter the esophagus. Okay? And once this food gets into the esophagus, it's just going to be transported into the stomach. The esophagus can contract, moves that bolus of food into the stomach. The stomach is a specialized compartment. It has smooth muscle, like all of the GI tract. It has these foldings that you see on the inside called rugae, and the rugae, and you see them right here as well. The rugae is just to increase surface area. It has a very high capacity, so it can store up to 2 liters of food and fluid. So very, very high capacity. It has 3 layers of smooth muscle, so it can contract in all directions. It contracts in this direction, and in an oblique direction to be able to better mix food with the chemicals inside the stomach. We'll talk about the chemicals in just a minute. So these chemicals, you guys know, is the gastric juice, right, which is hydrochloric acid. Hydrochloric acid is an acid, so it has a very low pH, 2 to 3, can get down to 1, so very acidic. And this hydrochloric acid helps denature proteins. Acidosis, remember, unfolds those proteins and makes them more prone to be digested by proteases. Proteases are enzymes that break down proteins, and pepsin is one of those enzymes in the stomach. Okay, so this acid not only helps denature those proteins so that pepsin has better access to it, it also activates pepsin, because pepsin is secreted by Schiff cells, and I'll talk about the cells in just a minute, in the form of pepsinogen, which is an inactive enzyme, and that acid activates it. Okay? So the acid is produced by these cells called parietal cells. You need to know that name. So parietal cells in the stomach produce hydrochloric acid. They also produce cofactor called intrinsic factor. So parietal cells produce the hydrochloric acid and also something called the intrinsic factor. The intrinsic factor helps protect vitamin B12 from getting broken down by the acid. Okay? And this vitamin B12, you use it to produce hemoglobin. So if you don't have that intrinsic factor, the person becomes anemic, meaning they're going to have less hemoglobin that they should. They cannot carry as much oxygen to tissue, so it's important for that. Schiff cells produce pepsinogen, which is the inactive form of pepsin. So pepsinogen gets converted to pepsin by the acid in the stomach. And what does pepsin do? It breaks down proteins. The other thing that Schiff cells produce are gastric lipase. Gastric lipase is an enzyme that breaks down lipids. Okay? Gastric refers to the stomach. So every time you see the word gastric, that refers to the stomach. So this is an enzyme that breaks down lipids in the stomach, and it's produced by Schiff cells. Now, in the stomach, it's very hard to digest lipids. Why? Have you guys ever poured, if you grab, and do this at home, grab a bowl of water and pour some oil in it. What happens to the oil? Does it mix with the water, or does it clump away from the water? Clumps away from the water, right? You put it on and they're going to create two layers. You're going to have the oil layer separated from the water layer. They don't like each other, okay? Lipids are hydrophobic. Phobia means don't like, okay? Don't like water, hydro, okay? So what happens in the stomach is that this solution that is produced by the parietal cells of hydrochloric acid is actually aqueous. It's actually watery. So lipids tend to form these gigantic clumps, right? Trying to have the least surface exposed to that water. So you're going to see the lipids clumping in the stomach. And if you have a large volume, you're going to have a very small surface area, right? So then it's not going to be very easy for gastric lipase to properly break down those lipids. There's going to be some lipid digestion, yes, but very little, just because it's hard to break down those big lumps of lipids. Does that make sense? Okay. So we have the parietal cells producing the acid. The acid is going to help us denature, basically unfold those proteins. And the acid is also going to help us activate pepsinogen into pepsin. Pepsinogen produced by the Schiff cells. Pepsin is going to break down those proteins. So digestion of proteins is happening in the stomach. Okay? The Schiff cells also produce gastric lipase. Some lipids are going to be broken down, but very little. The salivary amylase that started breaking down carbohydrates in your mouth gets denatured once it gets to the stomach. It cannot take the acid. So no carbohydrate digestion in the stomach. Okay? the acid is also going to defend against pathogens. A lot of the pathogens, the bad pathogens, right, that are going to, actually pathogens is bad. So all those bad, microbes that you ingest in food or fluids, and a lot of it comes in through the respiratory system, a ton of comes in through the digestive system. They're going to be killed by the acid in the stomach. So it acts as a defense mechanism. Some things can keep going through, but most things are not going to be able to pass that acidosis in the stomach. Okay, so I told you about the acid, right? So how is the stomach, the cells of the stomach, the cells that line the stomach, not digested or damaged by the acid? I mean, it's 2 to 3 pH. That is very, very acidotic, right? That should not only kill the pathogens, but should also damage the cells. Well, the stomach protects itself. It has these mucus cells, so the mucus cells are going to be located lining the lumen of the stomach. So you see them right here. And also these necks of the gastric pits, and these are all the other cells that produce the acid and the pepsinogen and so on. Actually, there are 6 types of cells, but I'm not going to talk about all of them in this class. So we've talked about Schiff cells and parietal cells. Now I'm going to talk about mucus cells. Mucus cells produce mucin. And mucin is going to be, it's basically a polysaccharide. So it's basically a bunch of sugars attached to proteins with high bicarbonate. And that's the key. So mucin creates these, here are the mucus cells, and here is the acid in the stomach, right here, pH of 2. And here we create that mucin. These cells, these mucus cells are going to secrete the mucin, and it's going to create a chemical barrier. So a chemical barrier between the hydrochloric acid and the cells. So the hydrochloric acid is here, this mucin is here, imagine it like a gel, right? And then here you have all of your cells. And this has high bicarbonate. And what does bicarbonate do? Bicarbonate buffers the acid. Remember, bicarbonate reacts with that hydrogen and forms carbonic acid. And if you do that, right, you're not going to have a very low pH because you're going to decrease the concentration of hydrogen ions. So this layer here, mucin, has a pH of about 7. It's neutral, right? And the pH of 2 or 3 in this gastric juice, in this hydrochloric acid, won't be able to damage the cells because you have this chemical barrier. This mucus is mucin really because it has high bicarbonate protecting all of the cells. So that's how we protect the cells. However, there are some times where people can get these either gastric reflux or ulcers. Ulcers, gastric reflux is going to be that, I'm sure you guys have felt at some point in your life heartburn. Yeah? So that sensation of burning, and sometimes it's so strong that you feel like you're going to throw up, that is gastric reflux. Okay, so you can have heartburn, which is mild, and you can have gastric reflux, which is a little stronger. If it doesn't get taken care of, then it can cause ulcers. Ulcers are like blisters in the lining of the GI tract. And you can have 3 types of ulcers. You can have esophageal, gastric, or duodenal, depending on where the location where they're happening. So let's look at that. Here you have the esophagus. Here you have the stomach. And here you have the first part of the small intestine. All right? And there are, as I told you, sphincters. Sphincters are the rings, the muscle rings that prevent contents to go from one compartment to the other one. So you need those rings are usually closed, and they open when you want that bolus of food to go from the esophagus into the stomach. So this is the lower esophageal sphincter, lower esophageal sphincter, separating the esophagus and the stomach. And here you have the pyloric sphincter, separating the stomach and the initial part of the small intestine, which is called the duodenum. That initial part of the small intestine is the duodenum. So here you have the pyloric sphincter, and they're always closed. Why? Because you don't want that acid from the stomach to go back to the esophagus or go into the small intestine all the time, okay? So normally what happens is when we have that bolus of food, these lower esophageal sphincter relaxes, allows the food into the stomach and immediately closes, right? The stomach has high capacity, 2 liters, right? So when the stomach contracts, this pyloric sphincter opens for a tiny little bit, like seconds, and you get a squirt of that gastric juice with the proteases and the food into the small intestine. The small intestine is very long, but it's not very wide. So it has very low capacity relative to the stomach. So you don't want this whole thing to dump into here. Because even though we do have bicarbonate, like a chemical barrier to protect the small intestine, and we'll talk about that in a minute, what produces that, if you dump the 2 liters into the intestine, that chemical barrier won't be able to protect it. And then you'll get those ulcers, okay? So this pyloric sphincter, just every time stomach contracts, it quickly opens, lets a little squirt of that gastric juice with the proteases and the food into the small intestine, and then it closes again. If these lower esophageal sphincter or the pyloric sphincter are not working properly, they remain open more time than they should, then you're going to get ulcers. And so if you get ulcers if the lower esophageal sphincter is the one that is not working, the lower esophageal sphincter is this one, okay? So if that one is not working and you have acid going backing into the esophagus doesn't have mucin, a chemical barrier. So you create those ulcers, and that is going to be an esophageal ulcer, okay? If you get to those blisters, I mean, you can have gastric reflux and it'll get treated and you'll be fine, but if you don't treat it, then you can get those esophageal ulcers. If the pyloric sphincter is not working properly, so the pyloric sphincter is this one right here, and too much, it remains open for too long, a lot of that gastric juice going into the small intestine, then you get duodenal ulcers. Duodenum is the first part of the small intestine. There's 3 parts of it. Duodenum is the first part of the small intestine. The other thing, and then if the stomach, for example, produces too much acid, so if those parietal cells are producing too much acid, that's going to overcome that chemical barrier, that mucin, so it's going to be too much acid. If this increases too much, then this barrier is not going to be enough to protect the cells. You can end up with gastric ulcers. So gastric ulcers are in the stomach. Okay. Overproduction of acid can also cause esophageal or duodenal ulcers, because then, even though the sphincters are working properly, if you have too much acid, too much of that acid is going to squirt into the duodenum or the esophagus, causing ulcers as well. Or if your stomach is not producing enough mucin, if those mucus cells are not being able to produce enough mucin. So you guys have taken Advil, right? Or ibuprofen. Yeah? For headaches or just when you have pain or swelling and so on. And you know how in the indications it tells you do not take it on an empty stomach? Usually you have to take it with food. Because ibuprofen actually decreases the production of mucin. And it's going to affect that chemical barrier. And so if you have less mucin, right, and because you're taking that medication, then that acid production just normal acid production can cause gastric ulcers. All right? And that's why you shouldn't exceed the 6 pills in 24 hours and so on, because then you can start having problems with the stomach. Okay. Then after the stomach, we go into the small intestine, okay? So the small intestine is where most of the digestion and absorption happen. Small intestine is where most of the breakdown of the food particles and most of it being absorbed into the blood is going to happen. So it's a super important part of your GI tract. In the small intestine, you're going to have digestion of everything: carbohydrates, proteins, and fats, or lipids. Okay? And this is going to be aided by enzymes that are secreted by the pancreas and the small intestine to digest carbohydrates and proteins. Also, enzymes from the pancreas are going to digest lipids, but we need help from the bile produced by the liver, as I'll show you in just a minute, to be able to digest all these macromolecules. So let's talk about carbohydrate digestion first. So carbohydrate, a lot of the calories that you ingest are going to be carbohydrates. I mean, we all love those carbs, right? Like bread, rice, cereals, we like them. So we ingest these big glucose polymers, right? Starch and glycogen, and we need to break them down. We need to break them down into monosaccharides. Monosaccharides are small enough that we can absorb them. So we're going to start breaking down those glucose polymers with amylase. Amylase is that enzyme that breaks down carbohydrates. That's going to start in the mouth with the salivary amylase. So the salivary amylase is going to start breaking these into disaccharides, like maltose, sucrose, and lactose. Remember, so in the mouth and in the esophagus, salivary amylase is working, is breaking down those carbohydrates. When we get to the stomach, salivary amylase gets denatured. It's a protein, right? So it cannot work anymore in that acid environment. So in the stomach, we don't have digestion of carbohydrates. But once we go into the small intestine, we're going to have the pancreas secrete into the small intestine pancreatic amylase. Again, so it's an amylase, but now it's produced by the pancreas, and it's going to act in the small intestine, in that first part, the duodenum of the small intestine. So it finishes breaking these glucose polymers that couldn't get broken down by the salivary amylase, because you really have very little time, right? That's why they tell you chew 30 sometimes, is because not only that mechanically breaks down the food more, but it gives more time for the salivary amylase to do something. Because once it gets into the esophagus, it's very quickly going to go into the stomach. So you want to give it time to be able to chemically and mechanically break down. But whatever didn't get break down by the time we get to the intestine, the small intestine is going to be broken down by that pancreatic amylase into disaccharides. Now, the disaccharides are going to be broken down further into monosaccharides by these enzymes, disaccaridases. And I'm probably pronouncing them wrong, but these ones are produced and secreted by the small intestine cells. The small intestine cells are called enterocytes. So enterocytes are going to produce disaccaridases that are going to break down those disaccharides into monosaccharides. And these ones are the ones that can be absorbed. They can cross the intestinal wall and move into the capillaries, therefore into the blood. So glucose, galactose, and fructose. Now protein. Protein digestion happens due to proteases, okay? So proteases, remember, are the enzymes. Are the enzymes that are going to break down proteins, okay? You have endo and exopeptidase. I don't want you guys to worry too much about it. endopeptidase, endo means inside, they're going to break down the protein into 2 smaller peptides. So they basically cut them in half. Whereas exopeptidase, what they do is they cut the terminal end. So they're going to cut this terminal end and produce an amino acid, this terminal end produce an amino acid, and then you end up with a small peptide here. It doesn't matter. Just know that proteases are the ones that break down proteins. And this is going to start happening in the stomach. You have that pepsin. And then it's going to also happen in the small intestine by enzymes secreted by the pancreas. And we'll talk about those in just a minute. Okay, I'll talk about lipids in just a minute because I need to introduce the bile for that. So I talked to you guys about the pancreas. And the pancreas has endocrine and exocrine secretions. So the endocrine secretions we'll study on Wednesday. Remember, Wednesday, we won't meet here. I'll give the lecture on Zoom. And it's going to be synchronous, right? So I'm going to be on Zoom teaching it, okay? It's not going to be a video. So endocrine secretions like insulin and glucagon, we'll talk about that later. These are hormones produced by the pancreas. I want to focus on the exocrine secretions today. So exocrine secretions are produced by 2 types of pancreatic cells. Here's the pancreas, and you have the duct cells. The duct cells are these ones right here. They basically make the wall of the pancreatic duct. Pancreatic duct, you see it right here. It's a duct that is going to, all of the secretions from the pancreas are going to go, exocrine secretions of the pancreas, I should say, are going to go through this pancreatic duct and empty into the small intestine, the duodenum, that initial part of the small intestine. But these duct cells that make the wall of the pancreatic duct secrete bicarbonate. And remember, bicarbonate buffers hydrogen ions. So that bicarbonate secreted here into the small intestine, they're made by these duct cells. It goes in the pancreatic duct, as you see right here, and empties in the small intestine. So that's going to create a chemical barrier that is going to protect the small intestine from the acid coming from the stomach. Okay? So equivalent to the mucin. But it's sodium bicarbonate, so it's a little bit different. Chemically, it's a little bit different, but it has high bicarbonate, which is what buffers those hydrogen ions. So it's important. If you don't have those duct cells, then you're going to end up with duodenal ulcers. And these duct cells are part of the pancreas. So people that have problems with the pancreas are not going to be able to protect very well that small intestine from the acid of the stomach. So see how important the pancreas is? The other types of exocrine cells are acinar cells. So acinar cells are these ones shown right here. And what they do is they produce these inactive enzymes called zymogens. Okay? So these enzymes are inactive. They're going to be enzymes that digest proteins, that digest lipids, and that digest carbohydrates. Okay? And why do we want them inactive? We want them inactive because we don't want them to start digesting things inside the pancreas. So they're produced by the acinar cells, secreted into the duct. You see the acinar cells here are connected to the pancreatic duct. And in that pancreatic duct, they get released into the small intestine, but as zymogens, as inactive enzymes. Once these pancreatic zymogens get dumped into the lumen of the small intestine. So this is the lumen of the small intestine. These are the enterocytes, or small intestine cells that make the wall of the small intestine. So here we have the zymogens. They get secreted by the pancreas, go in the pancreatic duct into the small intestine. And these enzymes produced called enteropeptidase. So other type of enzymes produced by the enterocytes, which are the small intestine cells, are going to convert trypsinogen into trypsin. And trypsin is the one that activates those zymogens into actual active enzymes. Okay? I don't need you guys to memorize these names, okay? But just know how this happens. All right? They get secreted as zymogens, and they get activated by trypsin. Trypsin is formed by converting trypsinogen into trypsin, given that we have these enteropeptidase. Okay? So once they get activated, they're going to help us digest proteins, lipids. So these two are lipids, these two are proteins. And one that I forgot to put here is going to be pancreatic amylase. And that one is going to do carbohydrate digestion. Okay? So what causes the pancreas to release these zymogens? When the small intestine distends, stretches because you have food in there, so that's going to cause the secretion of the zymogens. Chemoreceptors sensing food in the intestine, or signals from the nervous system or the hormones. So the nervous system and hormones regulate digestion.